Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasist from Department of Biotechnology and Bioinformatics, JP University of Information Technology, Himachal Pradesh. Today I am going to talk about module that is vesicle mediated transport, and this paper is placed in membrane biophysics. Biological membrane have lipids as a major component, and that sometimes raises a concern of transportation of hydrophilic moieties which cannot cross the hydrophobic barrier created by this lipid bilayer. However, exchange of bigger polar molecule or nutrients across the plasma membrane is mediated by two vesicle transportation processes. The current module deals with these special kind of transport which is carried out by the vesicle or membranous structures and numerous specialized intracellular compartments of the eukaryotic cell, with how molecules are moved rapidly and specifically between them in eukaryotic cells. Prime focus of this module is mainly on these vesicular transport process across plasma membrane. In view of this, following learning objectives are prepared for this module. Firstly, the introduction and need of vesicular transport. Then, different forms of vesicular transports. Thirdly, we will discuss about functional components of exocytosis transport. Then we will move towards structure and mechanism of action of endocytosis transport. And lastly, we will talk about molecular mechanism of receptor mediated endocytosis with its biological relevance. Biological membranes are external boundaries of cells that define external boundaries and regulate the exchange of essential components across these boundaries. The physical properties of phospholipid bilayer, such as flexible structure, selectively permeable, and self-sealing, allows cells to change their shape that is essential during cell division. Sealing properties of plasma membrane allow fusion of two membranes and become one, as well as fission of a single membrane result in two sealed compartments without leakage through cellular surfaces. This characteristic feature is utilized in transportation of soluble, insoluble, and large molecules across membrane by a process of vesicular transport. Exocytosis is the export of materials from inside of the cell via vesicles using secretory pathway, which involves the fusion of the membranes of secretory vesicles with the plasma membrane. It results in the release of the vesicle contents from the cell and also the delivery of the vesicle membrane proteins into the plasma membrane. Exocytosis is important in expulsion of waste material out of the cell and in the secretion of important cellular products such as digestive enzymes, secretory proteins, and hormones, etc. During exocytosis, the Golgi complex packages macromolecules such as proteins and polysaccharides into transport vesicles that further travel to and fuse with the plasma membrane. This fusion with the plasma membrane causes the vesicle to secrete their content out of the cell. Endocytosis is a form of active transport in which a cell uptakes materials from outside into the cell with the help of an energy-using process. Endocytosis, in general, the counterpart of exocytosis and used by majority of cells because most chemical substances important to them are large polar molecules that cannot pass through the hydrophobic plasma or cell membrane by passive transport. Cells use three major types of endocytosis processes, namely phagocytosis, macropinocytosis, and lastly, the receptor-mediated endocytosis. This figure shows a schematic representation of two vesicle-mediated processes that are endocytosis and exocytosis. In exocytosis, molecules are moving towards outside of the cell, while in case of endocytosis, hydrophilic molecules are captured into the membrane structures that we call them vesicles, and they are coming into the cell. The first stage of vesicular transport is the production of membrane-bound small transport vesicles. Small vesicles formation require deformation of the lipid bilayer, by which a goblet-shaped invagination of the membrane appears that will eventually be pinched off to form a vesicle. This process we generally call it as budding. The budding from the parental membrane and the formation of a new vesicle is driven by interaction and polymerization of soluble proteins. Vesicle formation is an energy-dependent process and is mediated by proteins such as pepsin, which are required specifically for budding. A formed vesicle carries the protein that were present in that stretch of membrane as well as the molecule that has to be transported. At last, fusion of vesicle occur with the membrane of target location, where the soluble molecule reaches its destination. The proteins that direct the targeting of the vesicles to the correct cellular location also mediate fusion and in some systems regulate the precise time at which fusion occurs. Several vesicles have a coat of protein surrounding their membrane and are therefore called as coated vesicles. The coat is acquired as the vesicle buds from the donor membrane and is shed before the vesicle fuses with the target membrane. There are three types of coated vesicles that help to localize biomolecules to a particular compartments. Number 1. Clathrin coated vesicles. Endocytic and secretory vesicles have clathrin as the most prominent protein in their coats, and these clathrin coated vesicles are responsible for transportation of proteins from the plasma membrane or the cell surface into the cell and the trans-Golgi networks to late endosomes. Number 2, they are the coat protein 1 coated vesicles, or we generally say these are COP1 coated vesicles. The vesicles involved in the transportation of proteins in the retrograde direction between Golgi cisternae and from the cis Golgi back to the rough endoplasmic reticulum. Number 3 are the COP2 coated vesicles. These vesicles are involved in trafficking from the rough endoplasmic reticulum towards the side of cis Golgi network. This figure shows all the three coated vesicles present in a cellular system, which are clathrin coated vesicles, COP1 coated vesicle, and COP2 vesicles. Clathrin coated vesicles are involved in the endosomal pathway and in transport of proteins from the trans-Golgi network, while COP1 coated vesicles are involved in the retrograde transport from the trans-Golgi apparatus back through the Golgi cisternae towards the endoplasmic reticulum. And lastly, the COP2 vesicles are required for transport from the endoplasmic reticulum towards the cis-Golgi system. Vesicles budding initiation require the recruitment of small G proteins to a region of membrane curvature, which then assemble the complex of core proteins and adapter proteins. GTP binding and hydrolysis control the formation of the vesicles. The G protein exchanges GDP for GTP, which is an active form, which can then insert spontaneously into lipid bilayers by means of a hydrophobic tail. The activity of G protein is controlled by the hydrolysis of GTP. Each vesicle contains members of GTPase superfamily for the exchange of GTP along with specific proteins. Both COP1 and clathrin vesicles having ADP ribosylation factors, that's we call the ARF factor, as GTP binding protein, whereas coat of COP2 vesicles contain related GTP binding protein known as SAR1. This figure shows an overview of a vesicle structure, which is consisting of G protein, a membrane receptor protein, membrane cargo protein, and adapter proteins. Small membrane G proteins attach to the membrane. They recruit coat proteins and adapter proteins. The cargo may be a soluble molecule that binds to the membrane receptor that are attached to the adapter within the coat. To retain essential components inside the cell, vesicles should be able to discriminate among soluble cargo protein and membrane proteins. Specific signal sequences, which are known as sorting signal, recognized by vesicles and hence localized in a particular cell compartment. Interactions occur between the cytosolic portions of integral membrane proteins and the vesicle coat the particular cargo protein inside the forming vesicle. Hence, curvature is added to the vesicle simultaneously, can also select which protein are admitted into the vesicle. This particular table shows the sorting signal sequences which are required to target specific transport vesicles. If you will find that if a protein is having a signal of KDEL, it means these signals are required for resident of endoplasmic reticulum and the receptors are KDL receptors which are present in cis-Golgi apparatus, and vesicles that incorporate signals bearing proteins are COP1 vesicles. Similarly, you will find if a sequence is present in a protein which is having a 2 lysines followed by 2 hydrophobic amino acids, then these are also resident of endoplasmic reticulum which are present in the cytosolic domain. In the table, you will find the mannose-6 phosphates are required for resident of lysosomal enzymes. Simultaneously, if you have leucine-leucine structures, these are resident of plasma membrane proteins in the cytosolic domain. Exocytosis is the vesicular transport process by which molecules are released to outside of the cell. Proteins are recruited to the plasma membrane as well as the release of secreted molecules into the extracellular matrix occur through exocytosis. In addition, higher eukaryotic cells communicate with each other via a variety of intracellular signaling molecules such as hormones, cytokines, and other small signaling molecules that are released from secretory vesicles. Moreover, exocytosis is responsible for the transmission of a signal from one cell to another, which occurs by the secretion of neurotransmitters into the synaptic cleft in response to the arrival of an action potential. Exocytosis may be differentiated into constitutive exocytosis and regulated exocytosis or secretion. Constitutive exocytosis is carried out by nearly all cells to transfer molecules from the Golgi apparatus to the plasma membrane of the cell. Constitutive exocytosis is not an unregulated process, and the rate of release of the molecule to the cell surface depend upon their rate of production, which is basically regulated by transcription and translation processes. While regulated exocytosis occur in response to a specific biochemical signal, which results the release of cytokines, hormones, neurotransmitters, and other small signaling molecules such as histamines. This figure shows a schematic representation of constitutive exocytosis and regulated exocytosis. In constitutive exocytosis, the process is going on without any signal, and the molecules will move from trans-Golgi apparatus towards the extracellular matrices. While in case of regulated secretion, a signal is required to fuse the secretory vesicle towards the outside of the extracellular matrix. Regulated secretion is typically controlled by external signals transduced via cell surface receptors. Molecules in the trans-Golgi networks are sorted into secretory vesicles either for regulated secretion if they contain appropriate signal sequences, or for constitutive secretion if they do not. For regulated secretion, proteins are first assembled in immature vesicles, and these vesicles mature as secretory vesicles near the plasma membrane. The release of insulin hormone by beta cells of pancreatic islet of Langerhans by increase in blood glucose level is an example of regulated secretion. Insulin is retained in secretory vesicles that are triggered to fuse with the plasma membrane in response to a rise in intracellular calcium concentration. A series of interactions occur which links the rise of glucose concentration in blood to the rate of insulin secretion. A rise in blood glucose causes the ATP-ADP ratio in the beta cells to rise, which closes the potassium channel in the plasma membrane. The resulting change in the beta cell's membrane potential opens calcium channels, causing a rise in intracellular calcium concentration and the secretion of insulin. The process in which cell internalize molecule from outside into its cytosol is termed as endocytosis. Endocytic mechanism control the lipid and protein composition of the plasma membrane, thereby regulating how cells interacting with their environment. Endocytosis describes the de novo production of internal membrane from the plasma membrane lipid bilayer. In doing so, plasma membrane lipids and integral proteins and extracellular fluid becomes fully internalized into the cell. Endocytosis is a key process in regulating processes like mitosis, antigen presentation, and cell migration. There are a number of endocytic mechanisms, each of which involves a unique proteins. The endocytic pathway comprises two distinct kinds of endosomes, precisely early endosomes and late endosomes. Material taken up by endocytosis passes from the early endosome to the late endosome, and from there may intersect with trafficking pathways from the Golgi apparatus, or may be directed to lysosomes or to the Golgi. The exact pathway depends on the cell and the material that has been internalized. Endocytic mechanisms are differentiated into three subcategories, namely macropinocytosis, phagocytosis, and receptor-mediated endocytosis. In macropinocytosis, small soluble molecules get the uptake in the form of vesicles, which are basically 0.5 to 5 micrometer in diameter, which usually occurs from highly ruffled regions of the plasma membrane. These vesicles are filled with a large volume of extracellular fluid and the molecules within it. No specificity is required in filling of the vesicle. These vesicles travel into the cytosol and get fused with endosomes and lysosomes in their later stages. While phagocytosis is an endocytic process in which cells bind and internalize large insoluble particles, which are basically 0.75 micrometer in diameter, like cell debris, microorganisms, and sometimes the apoptotic cells. Receptor-mediated endocytosis is an endocytic uptake of specific molecule from the external environment using a receptor, which generally present on the membrane surface and have specificity for the internalized molecule. In general, receptor-mediated endocytosis may occur by two different types, that are clathrin-mediated and cleavable endocytosis. Firstly, the clathrin-mediated endocytosis is mediated by the production of small, approximately 100 nanometer in diameter vesicles that contain a coat made up of cytosolic protein that we call clathrin. In this process, cargo recruited into the developing clathrin coated pits that we call CCPs and results in the formation of clathrin coated vesicles. Coated pits can concentrate large extracellular molecules that have different receptors responsible for the receptor-mediated endocytosis of ligands, for example, low-density lipoprotein, transferrin, growth factors, antibodies, and many others. Clathrin consists of a heavy chain and a light chain and successively assembled into polyhedral cage-like coat on the surface of the coated pit. The clathrin coat is made up of sub-assemblies, each consisting of three prolonged protein complexes, a triskelion, each leg of which is made up of one heavy and one light chain. The triskelion forms a lattice-like network of hexagons and pentagons which attaches to the membrane via an adapter protein, or that we call the AP complex. Adapter proteins bind both to the clathrin and to the integral membrane proteins, which are destined to be internalized and stimulate its assembly. Much more importantly, by binding to the molecules in the membrane of the vesicle, adapter proteins appear to be responsible for recognizing the appropriate cargo molecules. This figure shows a clathrin triskelion which forms the heavy chain and the light chain in form of hexagon, and the B structure shows a pentagon around the coated clathrin coated vesicle. Cavalier endocytosis and another common non-clathrin coated endocytotic process in smooth muscles, type 1 pneumocytes, fibroblasts, adipocytes, and endothelial cells. In this process, plasma membrane buds are present which consist of cholesterol binding protein, caveolin, that's VIP21, with a bilayer enriched of cholesterol and glycolipids. Caveolae are small, approximately 50 nanometer in diameter, flask-shaped pits in the membrane that resembles the shape of a cave, hence the name comes out. Extracellular molecules are taken up by specific receptors present in this cavalier endocytosis. Podocytosis is a kind of receptor-mediated endocytosis in which a vesicle bearing molecules of various sizes into the cell. However, material endocytosed by photocytosis is released into the cytosol, not into lysosomes or any other organelle as done in other endocytotic processes. Low-density lipoprotein acquisition by endocytic pathways. Low-density lipoprotein, that's LDL, consists of a spherical outer phospholipid shell having a single molecule of large ApoB100 protein and the core in which the cholesterol is packed in the form of cholesterol ester. Cell membrane contains surface receptors that specifically bind to ApoB100 protein and internalize LDL particles by receptor-mediated endocytosis. Cytosolic tail of the LDL receptors contain NPXY sorting signal for the binding with the AP2 complex that help in the incorporation of receptor ligand complex into developing endocytic vesicles. Dynamin mediated mechanisms are responsible for clathrin coated pits or buds containing LDL receptor complexes to pinched off. After internalization, the LDL particles are transported to endosomes and the acidic pH in the late endosome causes a conformation change in the LDL receptor that is responsible for the release of the bound LDL particle. The late endosome fuses with lysosomes and the protein and the lipid of the free LDL particle then broken down to their constituent parts by enzyme in the lysosomes. LDL receptors which dissociate from their ligand in the late endosomes recycle to the cell surface where at neutral pH of the exterior medium the receptor undergoes conformation changes so that it can bind another LDL particle. The LDL receptor makes one round trip into and out of the cell every 10 to 20 minutes for a total of several hundred trips in its 20-hour lifespan. Now, iron is also absorbed by the process of endocytosis. For that matter, we require a protein that we call the transferrin. Transferrin is a major glycoprotein in the blood that transport iron to all tissue cells from the liver and from the intestine. The apotransferrin, which is iron-free form, binds two ferric ions very tightly to form ferrotransferrin. Transferrin receptors present on the cell surface of all mammalian cells that bind ferrotransferrin at neutral pH. After binding of ferrotransferrin with receptors, these complexes concentrate in clathrin-coated pits and ferrotransferrin subjected to endocytosis. Change in the pH is critical for the functioning of transferrin endocytic pathway, which happens due to the fusion of vesicles with endosomes. At pH below 6 in late endosomes, the two bound ferric atoms dissociate from ferrotransferrin, reduced to ferrous by an unknown mechanism, and then they are exported into cytosol by an endosomal transporter specific for divalent metal ions. However, at the low pH of late endosomes, apotransferrin still remains bound to the transferrin receptor. After dissociation of the iron atoms, the receptor apotransferrin complex is recycled back towards the cell surface. Now, apotransferrin loosens its binding with the receptor at neutral pH and the bound apotransferrin dissociates from transferrin receptor at neutral pH of extracellular medium. The recycled receptor is then free to bind another molecule of ferrotransferrin and the released apotransferrin is carried in the bloodstream to the liver or intestine to be reloaded with iron. So students, let's now summarize what we have learned from this module. Firstly, sealing properties of plasma membrane allows fusion of two membranes and become one, as well as fission of single membrane results in two sealed compartments without any leakage through cellular processes. This characteristic feature is utilization in transportation of soluble, insoluble, and large molecules across membrane by a process that we call a vesicle transport. Molecules can be transported in vesicles across cell either in solution, in a non-specific manner, or by receptor-mediated mechanisms. Exocytosis is the export of materials from the inside of the cell via vesicles using secretory pathway, which involves the fusion of membranes of secretory vesicle with plasma membrane. Secretory vesicles stored secreted proteins and fuse with plasma membrane to release their contents. The formation of transport vesicle is initiated by small G proteins that insert into the donor membrane and assemble coat protein. The coat proteins are COP1, COP2, or clathrin, depending upon the pathway, and the coat includes adapter proteins that link the coat to the vesicle and its cargo. Epsin and dyneamines are involved in the budding processes. The vesicle cargo depends on the adapter proteins, sorting proteins, and receptors that are assembled in the vesicle. After vesicle have moved to the target membrane, GTP is hydrolyzed and the coat proteins depolymerizes to uncoat the vesicle. Endocytic mechanisms are differentiated into macropinocytosis, phagocytosis, or receptor-mediated endocytosis. In macrocytosis, small soluble molecules get uptake in the form of vesicles, which are generally a small diameter of 0.5 to 5 micrometer, which usually occur from highly ruffled regions of the plasma membrane. These vesicles are filled with a large volume of extracellular fluid and molecules within it. No specificity is required in filling these vesicles. These vesicles travel into cytosol and get fused with endosome and lysosome in later stages. Second mechanism is known as phagocytosis, in which cells bind and internalize large insoluble particles of having a size of 0.75 micrometer in diameter, and generally cell debris, microorganisms, and apoptotic cells are basically utilized in this phagocytosis. Lastly, the receptor-mediated endocytosis is the uptake of specific molecule from external medium using a specific receptor which generally present on the membrane surface and have the specificity for the molecule which has to be internalized. In general, receptor-mediated endocytosis may occur by two different types, that are clathrin-mediated or cleavage endocytosis. Cell membrane contains surface receptors that specifically bind to ApoB100 and internalize LDL particles by receptor-mediated endocytosis. In a similar way, iron can be internalized with the help of transferrin, which is a major glycoprotein in the blood that transport iron to all tissues, cells from liver and from the intestine by receptor-mediated endocytosis. Thank you.